start and stop on time. And hopefully people will be coming in. So I'm on mute for a couple minutes. Katrina, will the participants automatically come in? I don't have to let them in, do I?
Correct. They'll automatically come in.
Okay, thanks.
Mhm.
Welcome everyone so far. We are going to be getting started with the webinar in just a minute or two. I want to give people a couple of minutes to get into the Zoom meeting. So we will get started in a minute or two, but welcome. Okay, we're going to give it a couple more minutes before we start, just because I want to be sure we give everybody a chance. We did have quite a few registered, so hopefully they'll be coming in, but I want to give people a chance to get in. So we're going to give it another minute or so, and then we'll get started. Okay, I think in the interest of people's time, because I know people have schedules, we're going to go ahead and get started, and hopefully some other people will come in. But I'm Cindy Snyder. I'm the advanced genetics nurse practitioner that works at Georgia Core, overseeing our genetic screening program that we do in partnership with the Department of Public Health. But I also am the administrator and over the program for the Cancer Patient Navigators of Georgia. And as that person, we need to have quarterly webinars. So this is one of our quarterly webinars today. Everybody's going to remain muted on the presentation. And at the end of the survey, at the end of the webinar, the survey link will come up in the browser window for you to complete a survey to get your contact hour certificate. So just make sure that you keep a copy of that page once you complete the survey. If you have trouble with that, just email me. I've already put my email in the chat. So if you have a question or a problem related to that, let me know. You can post questions in the chat, or at the end of the presentation, if you raise your hand, I can allow you to speak. I have so much power. But anyways, I want to introduce our speaker today. It's Sandra Darilek, and she is a genetic counselor and associate professor for the Department of Molecular and Human Genetics at Baylor College of Medicine. And I heard Sandra give this presentation at the mid-year AONN conference, and I just thought it was so good, and I thought it was so useful that other people, navigators specifically, would get some benefit from hearing about pre-implantation genetic testing for hereditary cancer syndromes. So I'm going to mute myself and allow Sandra to continue the webinar. Thank you, Sandra.
Well, thank you, Cindy, for inviting me to give this talk. As Cindy said, I am a genetic counselor at Baylor College of Medicine. I work in reproductive genetics, so a little bit removed from oncology, but I always like to have the opportunity to make providers aware of reproductive options that are available for their patients. And I think this is especially true within the cancer realm for hereditary cancer syndromes. So I hope you find the informative and then also useful for the patients that you work with and that you serve. I don't have any disclosures other than the fact that I work for Baylor College of Medicine. I don't work for any genetic testing labs or any embryo testing labs or anything like that. So why would we be talking about reproductive medicine and embryo testing, particularly for healthcare providers that work with patients going through cancer evaluations, monitoring, treatment? Well, what we know is that genetic testing for cancer has really exploded in the last number of years. And as more individuals are actually undergoing genetic testing for either a personal history of cancer or family history of cancer, and then getting positive test results indicating that they do have inherited genetic cancer risk, they also hopefully are sharing this information with their family members, not only so that their family members know about these cancer risks, but also so that they can make the use of that information themselves. And so more people are also learning about their cancer risk because family members are having this testing. And more people are actually learning about this information when they're still at reproductive age, when they're still building their own families, in the middle of building families, or maybe even haven't started building families. And within this group of people that have this hereditary cancer risk, some of these individuals and some of these families might actually be interested in options to try to reduce the risk for that inherited cancer risk, or those particular predisposition syndromes in the next generation. They may want to try to prevent these risks from being passed to their children and ultimately their grandchildren. And so that's where this topic comes in, because this is a way of trying to reduce that risk for people who are building their families. So preimplantation genetic testing, or what we refer to as PGT for short, is an option that's used to reduce risk for a genetic disorder in pregnancy. So we don't, inherited cancer risk is certainly one option for this. It's been used much more often for inherited childhood conditions and those types of things. And so it's a way to reduce that risk. We never really talk about elimination of risk when we're basing things off of genetic testing, but it certainly can significantly reduce risk by doing a few things. You use in vitro fertilization, IVF, in combination with very specific genetic testing or screening, and then using the test results from that analysis that's done on embryos that are obtained from IVF and prioritizing embryos that do not have that genetic risk for transfer, ultimately, in the hopes of a healthy, unaffected pregnancy. I wanted to give you a little bit of background on terminology. It is a little bit of an alphabet soup of things that we use with the terminology. But first and foremost, PGT by definition from the American Society of Reproductive Medicine is a test that's performed on DNA from either oocytes in the form of polar bodies or embryos at either day 3, which is what we refer to as the cleavage stage of development, or day 5, the blastocyst day of development, which is the more typical time that we do this testing. And basically taking that DNA from either the oocytes or the embryo biopsy and then performing genetic testing. And that can be for a particular genetic anomaly that we're assessing for, or in some cases for things like HLA typing if we're looking for HLA matched siblings. There are several types of PGT. Probably the most common form of PGT is actually something we refer to as PGTA, which is the form of this testing that's used to test for general chromosome abnormalities, screening for things like Down syndrome. PGTM is the form that we do for inherited genetic risk and genetic disease. So that's what we'll be focusing on today. And then you've also got PGT-SR, which is for inherited chromosomal conditions, and then PGT-HLA, specifically for that HLA typing. So again, today, what I'll be focusing on is PGTM, because we'll be talking about that inherited genetic risk that is usually tied to a specific single gene in a particular family. So what does this entail? What does this require? Well, first and foremost, it requires IVF. And that's because that's what gets us the embryos that we can test. And then it also requires a few other things. So it requires the setup of a test specific to the couple to assess the embryos for the genetic change that we're worried about. It requires biopsy of the embryos that are obtained through IVF to actually get a sample to allow for testing. It requires freezing or cryopreservation of those embryos after they're biopsied so that they can be maintained until we get test results and still be available for transfer. And then once we have that biopsy, the actual analysis of those biopsied cells to give us the test result that we're interested in that can allow us to determine the status of a particular embryo. So IVF For those of you that don't live in the fertility world and specifically don't live in the assisted reproductive world, as a reminder, IVF is the technology that allows us to form embryos outside of the uterus. It is a process, and it can be a very long process for couples that undergo this. It's got some basic steps. The basic steps of an IVF cycle include ovarian stimulation, which is the period of time where a woman is given particular medication, usually daily injectable hormone protocols, where they are getting those medications so that their ovaries are actually maturing more than the typical one egg in a given cycle. The goal of IVF is to get multiple eggs from the ovaries at one time. Then retrieving those eggs from the ovaries, basically in a biopsy type procedure where we remove each of those matured eggs from the follicles within the ovaries. Once we have those eggs, they are evaluated, and then they go through a process of insemination with the partner sperm, which hopefully allows for successful fertilization, and then growth and culture of those fertilized eggs to become embryos. At the end of the day, once embryos are ready to be, are mature enough and have grown enough, they can be transferred. Usually we transfer day 5 or day 6 embryos from fresh IVF cycles. In the case of embryo testing with PGT, we actually freeze those embryos at day 5 or day 6 and hold them until transfer later, so that gives us the ability to do genetic testing. So the first step that kind of allows for embryo testing to actually happen is the process of an embryo biopsy. The image there on the screen gives you an idea of what an embryo at day 5 or day 6 looks like under a microscope. And it's typically at day 5 or day 6 in development that we are doing these biopsies, mainly because we've got a larger embryo, a bigger amount of cells within the embryo, and more specifically, because at this point, the embryo has started to divide itself into 2 major sections, and that can be a little bit hard to make out in this picture, but if you look closely here on the left, you'll see a group of cells, which is what we refer to as the inner cell mass. That's the part of the embryo that actually, if we get a successful ongoing pregnancy, becomes the baby in an ongoing pregnancy. And then that embryo is also lined by an outer layer of cells referred to as the trophectoderm that ultimately becomes the placenta in a pregnancy. So when embryo testing is done and when the biopsy is performed, we actually take the cells from the outer edge of the embryo, because they should be genetically identical to the inner cell mass, but there's also less risk to biopsying from the outer edge, less chance of doing damage to the embryo. So we do that biopsy by making an opening in the outer covering of the embryo, and they remove about 5 to 10 cells for testing. And that's important to kind of keep in mind. 5 to 10 cells is not a lot of DNA to work with. So the way we approach embryo testing all kind of hinges from the fact that we're getting a very small sample to work with in the first place. After the embryos have been actually biopsied, they are then frozen, and they remain frozen until test results are available and until we know which embryo we want to then thaw and then transfer to the uterus in the future. So at this point, embryos can stay frozen for a number of years and still be viable. So it does give couples, the ability to make reproductive decisions over the course of years and not have to, rush family building necessarily. Before you even get to the IVF part of things, though, you have to understand that there's an actual test setup process to this PGT. Unlike a lot of other genetic testing, IVF and PGT testing is very targeted, very family specific. And so prior to starting IVF, the lab that's actually going to be performing the testing for a given couple actually designs a genetic test specifically for that couple, for that patient, for the genetic change that we are trying to assess in that family. So what do they need for that? What do we need to know that they're going to need for our patients that are interested in this testing? Well, first and foremost, they need copies of genetic test results. So you actually have to have identified an inherited genetic variant that we're worried about passing on to the next generation. And we have to have documentation of what that genetic variant is. What gene is it in? What is the actual change that we're worried about? In addition to that, we actually need at the lab that's performing the testing a blood, saliva, or DNA source from the couple, as well as very likely from other members of the person who has the genetic variant, other members of their family who also carry that genetic variant. And you may be asking yourself, why do we need DNA from other family members? And I'm going to explain why that is in just a minute, because it all kind of goes into the way we do the approach for this testing. But we need DNA samples from the couple, from other family members, primarily that are positive for the variant as well. And most importantly, we need time. Time is something that, in my experience, working on the reproductive end, patients don't feel like they have enough of. And most patients that I talk to wanted to have started this process, yesterday. So it's very important for patients to know that test setup and this whole process does take place over a number of weeks. Test setup itself takes several weeks, and then the IVF process also takes a number of weeks. And I'll break down that timeline for you guys a little bit later. But before I dive into how we do the testing, I wanted to just take a quick second to talk about germline genetic variants versus somatic genetic variants, because I know in the cancer realm, there's a lot of somatic tissue testing, a lot of genetic somatic testing that's being done. And it's very important to be aware that what we need to know about for embryo testing are the germline variants. These are the genetic variants that are present in all of the cells of a person's body, including the germline, the eggs, and the sperm. And these are the variants that can be inherited. While somatic cancer testing can certainly find a number of variants and a number of genes in the cancer tissue that's being tested, sometimes those variants are only present in the affected cancer tissues. They are not necessarily always inherited variants. So if you start with somatic testing, we then need to make sure that we clarify which, if any, of those variants are actually inherited or germline variants. And it's those germline variants that we are interested in for embryo testing. So as far as the test process goes, the current test process for PGTM is very different from the way we really approach any other genetic testing. The current standard of practice is actually to use a process called haplotyping, which makes use of DNA markers, so polymorphic or markers that have some variation that are closely linked to the gene of interest or actually found within the gene of interest, and tracking those marker patterns as opposed to relying solely on the presence or the absence of a particular variant. The way I like to explain this to patients, and I'm going to walk you through it here in the next slide step by step, is for a patient that carries a genetic variant, inside that gene, they carry a genetic change. So they usually have two copies of the gene, one copy that has no variant in it and works perfectly fine, and a second copy that has that genetic variant. Well, also around that gene, they have these DNA marker patterns that are benign changes in the DNA code that we all have. But these marker patterns are incredibly unique. They are very unique to our families. These marker patterns are the same things that are used for things like forensic DNA testing. So when you've got a crime scene and there's a sample at the crime scene and you're trying to match that to a suspect or to a victim, often what they're using are these DNA marker patterns that are person and family unique and specific to match those things. Well, for years in genetic testing, especially with gene discovery, we were also using these marker patterns to track genetic variants through multiple generations of families. And so we go back to that technology when we're doing this embryo testing. And it all comes from the fact that we're dealing with a really small amount of DNA. And in the past, when we only relied on looking for the presence or the absence of a particular variant, we sometimes got it wrong. We got a false negative result, an embryo was transferred and ultimately still had that genetic variant. So as an approach to try to maximize accuracy of these test results, because if a couple is going to go through all of the hoops of IVF and embryo testing to get to the point of transferring an embryo they think does not have the genetic risk they're worried about, we want to make sure that accuracy is as high as possible. And we can get to 98% accuracy by using this particular approach. So how does it work? So haplotyping or linkage basically starts with the patient that we're interested in. So let's say, for example, you have a patient, she's 33, she tested positive for a BRCA1 variant, and she's worried about passing this on to her children and wants to try to prevent that. Well, what will happen is that the embryo testing lab will want her test results, so they know what that BRCA1 variant is. But what they're also going to request is a DNA sample from her, so they can figure out what the marker pattern is that she carries around her two BRCA1 genes. So again, she gets one marker pattern from her mom, one marker pattern from her dad, one travels with each of those BRCA1 genes. So for example, she could have a marker pattern that's ABDF from one parent and 2478 from another parent. Now that's good information to have, but what it doesn't tell us from that DNA from the patient alone is which of those two marker patterns is the one traveling with the BRCA variant that we're worried about. So that's where the need for DNA from other family members, particularly family members that have the same variant, comes in. So let's say she has a father who also carries that same variant. What the genetic testing lab is going to request is DNA from him as well. And what that's going to allow us to figure out is which marker pattern are we worried about. So when we get DNA from the parents of a patient, especially from the parent that is positive for the variant, what we do is we look at what they have in common. They both have the genetic variant that we're worried about. And in this case, they both have the 2478 marker pattern. That tells us which marker pattern we need to be worried about when we see it in the embryos. So what the lab will also do is get a DNA sample from her partner or from the source of the sperm in this case, so that we know what their marker pattern is. And they will use all of that information to determine the status of the embryos. So each embryo should get two marker patterns, one from the egg, one from the sperm. Any embryo that does not get the 2478 pattern from our patient is expected to be unaffected and to not have that inherited cancer risk. Those would be embryos we would prioritize for transfer. Any embryo that does get that 2478 marker pattern is expected to be affected. So they have that same cancer risk as our patient has ultimately. And those would be the embryos that are lower priority or not transferred. The accuracy of the testing comes in from the fact that we want to make sure that we see two marker patterns, one from the egg and one from the sperm. Because the reason we used to get misdiagnoses by focusing on the variant alone was the fact that the first step with any genetic testing, particularly with genetic testing that starts with a small amount of DNA, is a process called amplification. We actually make copies of the DNA that we get from the sample, so we have a larger sample to work with. That's incredibly important when you're starting with something like 5 to 10 cells. Well, unfortunately, sometimes when you're doing that amplification process, one of the two copies of the gene that you're trying to assess actually doesn't amplify. You don't get additional copies in to actually look at. And what will sometimes happen is that copy of the gene will do what we call drop out. It'll drop out of the analysis and it won't be present in the actual assessment. Well, in the past, when we only looked for the presence or the absence of a variant, when we didn't see it, the assumption was it wasn't present in that sample, it wasn't present in the embryo. But 5 to 10% of the time, that was an incorrect assumption. And we still had embryos that ended up being positive and pregnancies that were positive for those variants. And what had actually happened is one copy of the gene amplified and showed up in the analysis. But what we didn't realize is the copy of the gene that we actually needed to see didn't. And so while we got a negative test result, it wasn't because the embryo wasn't negative, or it wasn't because the embryo was negative. It was because the embryo that sample didn't show both copies of the gene. So this linkage approach actually catches that scenario. If only one copy of the gene shows up in the analysis, we only see one marker pattern. That is what we term an indeterminate result. And if that marker pattern is from the partner who does not carry the genetic variant that we're worried about, we know that we don't have all the information we need to make a call on that embryo. That prevents us from making the misdiagnoses that have happened in the past. That gets us up to the 98% accuracy that we can typically get with this testing. I know that's a complicated scenario, and I typically walk patients through this so that they can wrap their minds around it, but that's why this takes so long, and that's why we need DNA from other family members as well. So what's the actual timeline? So the timeline for IVF with PGT, and this is important if patients are interested in it, kind of breaks down this way. Most labs need about 4 to 8 weeks for the actual test setup to get the samples from the patient, from their partner, from other family members, and do all of the setup that's required. At the same time, the fertility specialist needs about that same window of time, 4 to 8 weeks, to do baseline evaluations, blood work, imaging of the uterus, the fallopian tubes, sperm analysis, to make sure all of that is going well. Those two things can be done simultaneously. Once you get past that point, there is and everything's ready to go as far as embryo testing goes. There's typically a 3 to 4 week period of time where a woman is put on birth control prior to the IVF, prior to the ovarian stimulation, which to a lot of patients seems counterintuitive. If they're trying to get pregnant, why would they need to be on birth control? But IVF is all about controlling timing, and this allows us to control timing. After that, there's about 2 weeks for ovarian stimulation, about a 1 week period from the time we retrieve the eggs to growing the embryos and biopsying them and then freezing those embryos. And then about a 1 to 2 week period of time to receive the test results. Once we have test results, embryos are typically transferred within 4 to 8 weeks after the results are received, if the couple's ready for transfer at that time. So if everything goes according to plan, no hiccups, no obstacles, and we go straight through, a patient's usually looking at 15 to 25 weeks from the start of this process to the transfer of an embryo. So that's about 4 to 6 months period of time that they need to be aware of and planning for. So as a provider, as a healthcare provider, what do you need to know about PGT? You need to know that PGT can be performed for most hereditary cancer syndromes if the germline genetic variant has been identified. There are exceptions. There are some variants that are incredibly difficult to test for. There are some families where we don't have enough information from other family members to do the setup we need to do, but in most cases, it is an available option. You need to know that testing is performed differently through this linkage analysis or haplotyping approach. And as a result, that requires several weeks for setup and typically samples from multiple family members and also test results on other family members. You need to understand that PGT does not replace prenatal testing or postnatal testing. While the accuracy is very high, we do still recommend confirmation either during a pregnancy or after a child is born of the negative test results, just for absolute reassurance. There is a risk of that embryo biopsy. While we do not see an increased risk for birth defects after biopsies are performed, there is a chance that an embryo won't survive following the biopsy, meaning that it won't continue to grow, or once it's thawed, it won't successfully implant. That risk is relatively small, in the range of 5%, but it is something that patients and providers should be aware of. You also need to be aware that cost and insurance is probably the biggest obstacle to this for most patients. Very often we are writing letters to insurance indicating the reason for why a patient is considering this. There are some funding sources that are available to families and couples that are trying to go through IVF for genetic testing reasons. What do patients need to think about? And I think this is important, especially when you're kind of giving the patients information about this. Things that they need to think about and be aware of is that, first and foremost, IVF and embryo testing doesn't guarantee a pregnancy. Unfortunately, while IVF can be very successful, there are a lot of couples that don't have successful IVF cycles. Also, IVF with PGT, while it does significantly reduce the risk for a genetic condition, it doesn't eliminate that risk, and we still do recommend prenatal testing or postnatal testing for confirmation. IVF with PGT does not test for all genetic conditions, and it does not test for all cancer risk. It is an incredibly targeted test. We are usually testing for one or two things, so we're not assessing for everything. It doesn't give us anywhere near the information that a prenatal test can give us, as far as the breadth of information you can get, or postnatal testing. But it does tell us what we're worried about that's running through a family. Patients need to think about whether they're comfortable with IVF in the first place and the testing of embryos. So that entire process, as well as the costs that go into that. And that includes not just the financial costs that most people automatically think about, but the physical and the psychological and the emotional cost and the toll that IVF can take. Because IVF is not a guarantee, it can become a very frustrating process if you don't get the number of embryos that you're hoping for, or you don't get the test results on the embryos that you're hoping for, or if those transfers are ultimately not successful. So couples really need to think about if they're ready for that process itself. They also need to think about whether they're comfortable with the creation and testing of embryos. Does that fall within their own value system, moral system? You know, how do they see embryos? They need to think about what they're going to do with embryos that are expected to be at risk for the cancer syndrome. As you guys are well aware, if you carry a genetic cancer variant, while that does put you at increased risk for cancer, it doesn't mean that you're guaranteed to develop cancer. And the same goes for the children of people who carry those changes. Just because they have the risk doesn't mean that they're going to get cancer. So they need to decide how they feel about those embryos that actually do test positive, and are they comfortable with discarding those embryos ultimately, or are there any situations where they would still consider transfer of those embryos? They need to think about what they're going to do if they have no embryos without the genetic variant. You know, while we're usually dealing with 50-50 odds, 50% chance that you're going to pass on the variant, 50% chance you're not, it's a flip of the coin, and you certainly can get a group of embryos that are all positive for a variant when you were only expecting half of them. It's like doing a coin toss and getting 4 heads in a row. So they need to think about what that could mean if they're in that scenario. And they also need to think about what they would do with the remaining embryos once they've completed their family. So sometimes people do very well with IVF, get a number of embryos, a number of negative tested embryos, but only want 2 kids or only want 3 kids. And so what happens if you are lucky enough to have extra embryos? What do you do with those? Do you put those up for adoption? There is such a thing as embryo adoption. Do you just discard those embryos? Do you donate to research? What are the options that are currently available, and what do you feel comfortable with? I know, especially from changes over the summer in our political environment, there's a lot of concern within the reproductive world of what changing laws are going to mean for what decisions patients get to make about their embryos, and whether the regulations that are going into place for pregnancy and pregnancy termination are going to extend to what you can do as far as discarding and transfer of embryos. And to be completely honest, we're still trying to figure that out. We're not quite sure where things are going to go in this realm. What I can say is, as of right now, today, no states have laws in place that dictate that you cannot, transfer certain embryos or have to transfer all of your embryos. There are no laws on the books currently that say that. Most states do not have laws right now that regulate disposition of embryos, and that is still a decision that is up to the provider and their patient. The only state that I am aware of that has any laws on the books that do affect what happens to embryos is Louisiana. Louisiana has actually had a law on the books for a number of years that says you cannot discard any embryo. I'm not quite sure what those clinics do with those embryos with that law in place, but patients still have the choice of what they want to transfer and don't want to transfer. So this is still an evolving situation, and it certainly is a big concern for both fertility providers as well as the patients that they serve. Another concern, particularly for patients, is the cost. And like I said, cost is usually the biggest obstacle to IVF and embryo testing for most families. An IVF cycle ballpark runs about $10,000 to $15,000. The medications run about $3,000 to $5,000. And then the embryo testing adds an additional $3,000 to $10,000, depending on the complexity and the number of embryos. The freezing and storage of embryos is about $500 per year on average. These are ballparks. Costs do vary from clinic to clinic, and also there are regional differences in cost. To be quite honest, IVF is very expensive in the US compared to other countries. I have certainly worked with families who have gone and done IVF in other countries because it was significantly less expensive in other countries. And insurance coverage for IVF is something that widely varies. It varies between different insurance companies. It even varies within the policies within the same insurance company. And it varies very much from state to state. So it's something that we tend to help patients try to figure out whether they've got coverage. Do they have coverage for IVF? Do they have coverage for embryo testing? What are the criteria that they have to meet to be eligible for that coverage? As I mentioned earlier, there are some financial assistance options for patients who are wanting to build their families through IVF, and particularly ones that are doing genetic testing as part of that. They're available through grants and loans. Resolve, which is a big infertility support group, has a beautiful website of resources that gives patients a lot of resources about how to make fertility treatments affordable. There are other groups like Fertile Thoughts and Fertility IQ that also have links to grants and charities and scholarships for couples and patients and trying to do this. From a cancer-specific perspective, there are some fertility preservation and family building grants. The Alliance for Fertility Preservation has links to some of these. And I believe that some of the cancer-specific support groups may also have resources about these. So breast cancer support groups, colon cancer support groups, specific genetic variant support groups as well. I always think it's important, being a reproductive genetic counselor, that people understand that IVF and PGT isn't the only option out there. So particularly when you've got patients who they themselves have cancer diagnosis, I think fertility preservation options are incredibly important to be discussed when people are of reproductive age and have cancer risk or cancer diagnoses. They need to know about egg banking, sperm banking, at least know that it's out there, whether they want to take advantage of that or not. I've worked with too many patients who wish they had known about that ahead of time. And sometimes time is not, in our favor, and we just don't have time to make use of those options, but sometimes we can. I think people also need to know that they can make use of donor eggs, donor sperm, and also donor embryos to build their family. Donors obviously reduce the genetic risk if we're dealing with an inherited risk. Donors are also really good options for patients who, particularly women who want to carry a pregnancy, but maybe their egg reserve was affected by chemotherapy and those types of things. And I also feel very strongly that adoption is not an option that's talked about enough as far as family building goes, but it is a very viable and a very important option for people to be aware of. So how do you find IVF and PGT resources where you are? Well, one good resource is to talk to your local genetic counselors, particularly your counselors that work in the prenatal realm, or if you have any that work specifically in the fertility realm. Fertility clinics, your IVF clinics are a good resource as well. They certainly can tell you if they offer embryo testing for inherited genetic disease, and a lot of them do at this point. I always think it's a good idea, being a genetic counselor, that you refer your patient to a consultation with a genetic counselor, just so you can talk through the ins and outs of that in more detail. If everything they've been hearing about so far is their cancer genetic risk and what that means as far as things go in their cancer realm and their own health, the reproductive conversation can be very different for them. And so sometimes it's helpful to have somebody to guide them through that discussion. Bottom line, the things I hope you take away from this is that PGT embryo testing is an option that's available for hereditary cancer syndromes, not just for pediatric genetic disease. It is a multi-step process, and it definitely is. It's not a quick fix for anything, but it is something that can make a big difference to some of these families. PGT does reduce, but we never say it eliminates risk. And, you guys being healthcare providers know we never give zero odds for really anything. And, last but not least, PGT is a good option for some patients. It's not right for everyone. So it's not going to be the best option for everybody, but for some people and for the people that would seriously consider it, I think it's important that they are aware that it's available and out there. And with that, I wanted to provide you my contact information. I'm always available to answer questions about anything that's happening in this realm. And I'm happy to answer any questions that anybody might have.
Thank you so much, Sandra. I just really find this, I know I might be kind of a genetic nerd, but I do find this quite interesting, because I have told many people about PGT as an option, but when you don't really know what the process is, it's different when you learn that it is definitely a process. And I think the biggest thing I take away from this, again, is that there is confirmation testing that needs to be done after the baby is born. So I think that's really important for people to understand.
I mean, I think it's important, the risk reduction is huge, so you're going very much from the perspective of going from a 50-50 chance of your child having this to pretty much just confirming a negative is what we expect. So mentally and psychologically, that can be huge, for parents, but it's not, it's not a guarantee. And there's always the chance for error, unfortunately.
Right. And I also found it very interesting when you were talking about the process where they're looking for the SNPs, if you will, the single nucleotide polymorphisms around the pathogenic variant, and how unique that is to the individual. And that's really the more accurate way of doing it versus just looking if they have it or not. I thought that was very interesting too.
Yeah. And even when I educate a lot of people, I educate a lot of our genetics residents and fellows here at Baylor, and that linkage approach is not a mainstream kind of test methodology anymore. It used to be. So even for people in genetics, when we talk about this is how we do this approach on embryos, it's kind of a new way of thinking for them, but the rationale for doing it is huge to get that accuracy.
Yeah. And also the fact that I guess for the most part, you're trying to biopsy those cells that do turn into the placenta. Is that basically?
Yeah.
Basically.
So in a perfect world, if we felt it was safe enough, we'd obviously want to get those cells from the inner cell mass that are the most direct representation of what's ultimately going to be the fetus and the baby. But we don't want, the embryo is so small, it's about 100 cells at that time. We really don't want to cause a problem that wasn't there to begin with. So we rely on those outer cells. With an inherited genetic condition, I think we feel very confident that those cells should be identical matches. Now, there are exceptions to that. From a chromosome perspective, that's a whole different story. And I do a whole different kind of counseling for other types of embryo testing. But for these inherited conditions, we can feel pretty confident that those outer cells, even though they're ultimately going to be placental cells, are pretty identical matches to what that inner cell mass in the baby would look like.
And if anybody, I have been saying post a question in the chat, but sometimes on the webinar, the chat is disabled. So you might try using the Q&A. I don't know. Let's see here. The chat is now available. So, okay. Thank you, Katrina. That's why we need a second chair. So there may be a question coming up in the chat here in just a second. And I was going to say, I had something else on my mind before that kind of popped up, but so do you have any concerns? I know you mentioned it during the presentation. Do you have any concerns with the current environment related to women's health? Let's put it that way.
Yeah, so I mean, I practice in Texas, so yeah. I think what is favorable, at least right now, is there have been no trigger laws on the books that seem to the American Society of Reproductive Medicine has their legal team actually keeping an eye on this, you know, month to month, and updating the larger group of us. And right now, there are no trigger laws that really affect embryo disposition. Whether that's going to change, I don't know. You know, my hope is, and this is probably naive in the political environment that our country is currently in, my hope is that, you know, our legislators wouldn't want to prevent people from building their families. And, you know, IVF doesn't just affect people who have genetic disease running through their families. These are people who have no other way, you know, who have tried for years to have children and unsuccessfully are having children. And so I hope that, you know, they don't want to put limits on the ability of women and couples to, you know, build families. But honestly, I don't know. I don't know what it's going to look like. And I have a lot of patients who are in the middle of IVF cycles who are terrified about what they're going to be able, you know, what decisions they're going to have to make about embryos they already have. So
I can't even imagine having to make those tough decisions. And like you said, you know, the process being 4 to 6 months, I mean, there's always things that can happen in that time. So Cheryl had a question. I don't know if you can see the question in the chat.
So I think the question is, how often are embryos frozen during most IVF procedures? You seem to dispel concern about freezing embryos that is necessary for PGT. So currently, actually, frozen embryo transfer is the primary mode of transfer. Most IVF cycles are no longer fresh transfer cycles. They are frozen transfer cycles. And that's because, for a few different reasons, but we have found that frozen embryo transfers have pretty much the same success as far as implantation goes for fresh embryo transfers. And the belief is that giving the woman's body a chance to rest from the significant uptick of hormones that they are given during the actual ovulation stimulation actually does better for implantation. That pause between the actual retrieval, letting the body get back to baseline, and then doing a frozen transfer later gives the body and the uterus a more natural environment to work with. We've gotten better at freezing embryos, which I think is why these embryos survive better. They now do vitrification, so flash freezing, which gives them they basically freeze them down very quickly. And so we don't have as much crystal development within the embryos, which is what did the damage in the past. They've been freezing embryos in a variety of ways for about 3 decades, and we don't see a significant increase in the outcomes for birth defects for pregnancies that were frozen embryo transfers versus IVF fresh embryo transfers. So I think the data is very reassuring that frozen embryos don't have significantly higher risk than any other IVF embryos.
That's good. Thank you for that question. I really am so glad we're recording this, because we did have people registered, but I mean, things happen, life happens, but having this recording available, and I hope you're okay with us doing that. But
Yeah, absolutely.
because I think it would be very useful for, potentially even people in the public, to learn about this, but for sure, cancer patient navigators.
Yeah.
To know this and to have an option to be able to view the recording later.
Yeah.
So I really appreciate your willingness to do this today.
Well, yeah, and I think part of the battle is just getting providers to remember that this is available. I mean, you've got so much that you're talking to for patients that are going through cancer treatment, cancer evaluation. They've got bigger fish to fry most of the time, but when they're of reproductive age, I think having this as part of the conversation so that they can decide if they need to explore this when they have more options available to them than after treatment, when maybe their options are significantly more limited. At least they don't feel like they missed the boat on knowing that. And I think, just giving them the power to decide what they're more worried about in that time, I think can, for patients that feel like, they don't have control over much at that time, giving them any bit of control back can be really helpful.
Oh yeah, definitely. I was going to give anybody a chance to either put a question in the chat or raise your hand, and I can try. Okay, here we got another one from Janice. Can you see that?
Yeah,
Sandra?
so basically the question is, what is the percentage rate of embryos dying after you do the procedure or take the cells for testing? So that's a really good question. So from a biopsy perspective, it's estimated that you might lose about 5% of embryos, that the loss is related to the biopsy itself. So certainly not a, negligible risk, but not a high risk that goes along with that. If you're thinking about like freezing embryos, because our freezing processes have gotten better at this point, probably 80–90% of embryos survive freeze and thaw. That can be different from group to group because, processes are a little bit different in different IVF clinics, but the processes by and large majority of the embryos actually do survive being frozen. And then the next question, it looks like, is there a relationship between the duration of time an embryo is frozen and poor outcomes? Examples, birth defects, non-implantation, etc. Actually, no, we're not necessarily seeing the duration of the freezing being an issue. I have certainly had patients who have had embryos frozen for 4 or 5 years and had success with transfer. I think it comes back more to the quality of that embryo before it was frozen to begin with. So what did it look like morphology wise, genetically? What did it look like? Not, maybe not just from an inherited cancer risk perspective, if that's what we're testing for, but maybe from a chromosome perspective, which are the bigger, drivers of miscarriage and failed implantation and those types of things. So time being frozen doesn't seem to be a big variable as far as success for implantation later.
Great. Thanks for that question.
And I think for a lot of couples, that's important because it gives that, it makes them feel like they don't have to like rush these decisions. So.
Right. Which is important because like you said, they got bigger fish to fry if they're getting cancer treatment.
Absolutely.
So, definitely. in the last couple minutes that we have, I just want to remind everybody that at the end, when I end the webinar, a browser window will come up with your survey. You can fill that out and get your contact hour certificate. Just make sure you save that. If you do have any questions about that process, you can email me. I put my email in the chat, csnyder, SNYDER @georgiacorps.org. And I just want to again thank Sandra for her time today for this topic. It's really important. And I'm just really glad we got to record it. And any last words, Sandra?
No, I mean, thank you again for inviting me. This is a topic dear to my heart. I want people to know what their genetic testing options are, no matter whether they would take advantage of them or not. And so I think the more we can get the word out, the more that patients benefit from that. So thank you for having me.
Okay, last chance for any questions. Everybody's saying thank you. Thank you for attending. And we'll end the webinar now. And you have a great rest of the day, Sandra.
All right. Thank you so
Everybody
much.
else. Okay. Thank you.
Bye.
Bye-bye.